Well, let's look now in a little bit more detail at the molecular level as to how this happens. Well, we have our 11 cis retinal that is in the center of the opsin, and when light strikes this molecule, there's a rotational twist between carbon atoms 11 and 12. That converts the 11 cis retinal into an all-trans configuration. So you may remember a bit of organic chemistry there, the difference between a cis and a trans. Well, this conformational shift is what leads to the activation of the metabotropic element of the opsin protein. So with that in mind, here is light striking our photopigment, and the twist that we see in that retinal molecule leads to the activation of a set of G proteins that are affiliated with that opsin. Those G proteins are called transducin, and one consequence of activating these G proteins is that a phosphodiesterase enzyme is turned on, and that phosphodiesterase cleaves the cyclic structure of the cyclic GMP molecule. And as a consequence then, the cyclic GMP levels fall, and the channel goes from its open configuration to its closed configuration. Rather remarkable that all of this happens as quickly as it does. Well, in neural terms, it actually does take a bit of time, which contributes to a bit of the lag in the operation of the visual system. The visual system is actually one of our slowest sensory systems, in large measure because of the time necessary for this phototransduction cascade to play out. Well, now let's consider some features of rods and cones that help to differentiate their contributions to vision. Rods are extremely sensitive to light, for reasons that we'll mention in just a moment. Cones, on the other hand, are relatively insensitive to light. Vision, when driven by rods, is characterized by low spatial resolution, and this pertains to the wiring of rods to ganglion cells in the retina. Cones, on the other hand, are associated with very high spatial resolution. Putting these facts together, rods are specialized for very low light levels. We call this scotopic vision, whereas cones are specialized for very bright conditions, such as this beautiful late afternoon here in North Carolina where I'm talking to you today. So this photopic vision enables color vision, since color vision requires the activation of cones. Well, let's consider some of the physiology and anatomy of our rods and cones that helps to explain these features. If we look at the response of an individual rod and a cone to the onset of light, what we find is that in a rod, there's a graded response, graded by the intensity of the light stimulus, but there's also a very sustained response that adapts relatively slowly. Now contrast that to the responses of the cone, which also responds in a graded fashion, but the adaptation is very quick. So the metabolic mechanisms that are responsible for adaptation within the outer segment of these cells operate on different timescales, with a much more sustained response seen in rods relative to cones. Another specialization pertains to the wiring of rods and cones to the ganglion cells, and this is shown rather schematically here in this figure. Notice that about 15 to 30 rods connect up with a single bipolar cell. Contrast that to the cone system, given this one-to-one relationship, and this cone bipolar cell can be sensitive to a much more narrow spatial channel as photons interact with the retina. We can consider the specializations of rods and cones in terms of their physiology and their anatomy and begin to understand how we can be sensitive to light over the incredible range of luminance that's present in our environment. We're sensitive to more than a 12 log unit range in luminance. We can define an absolute threshold for vision, and this occurs under extremely dark conditions. Imagine being in the darkest woods in the darkest night, and your way illuminated only by starlight. What little vision that's possible under those conditions is due entirely to the operation of our rod system. This is called scotopic vision. Now, in conditions such as where I find myself now, looking into the setting sun, there is considerably more photons in this environment right now. And I suspect that I'm somewhere around this amount of illumination now, where our cones are responsible for the vision that I see. The rod system has completely saturated at this point. So all that I see in my world around me at this moment is due to the activation of cones in my retinas. So these kinds of conditions, when cone vision dominates, are called photopic. Now there is a transitional region in the middle called mesopic illumination, and this would be characterized by, let's say, being out in the woods, and in addition to starlight now there's moonlight, maybe about a quarter moon or more. That would get us in this range where we are beginning to approach photopic conditions that are possible when there's nearly a full moon. So this operating range is incredible, and it is a real feat of evolution that we can remain sensitive to the dynamics of light in our environment across such a huge range in the physical world. And that has to do with the responses of the neural elements and how they interact with one another to actually produce the signals that are conveyed from the retina to the brain. Well, one final specialization to consider is the distribution of rods and cones across the retina. Well, I've already mentioned earlier that in the center of our vision we have a very high density of cones, and here's what this looks like. So what we have in the plot here down below is the distribution of cones, shown in green and very low levels in the peripheral parts of the retina. But as we approach the very center of our gaze, we find this very interesting specialization in the retina called the fovea. And in the fovea, we have the complete exclusion of rods. And what we see is a very high density packing of cones with minimal diameter to the outer segments of those cones, making them as sensitive as they possibly can be with respect to the spatial arrangement of the distribution of photons that might be striking that retina. As the cone system gets further removed from that center of vision, notice that there are far fewer cones that are present, and they tend to be much larger as we get out. Now contrast that with the rod system, and what we find are that the concentration of rods increases as we get near the center of the retina, but then it falls precipitously as we approach the fovea to a total area of exclusion. And then it rises again, only to trail off as we approach the peripheral parts of the retina, where the density of all photoreceptors begins to trail. So this fovea then is quite an interesting spot in the retina. Let's take a closer look at it. If we looked at the architecture of the connections between photoreceptors and ganglion cells in the fovea, what we discover is that there is a pit in the middle of our fovea. And fovea itself means pit, but that central region is called the foveola. And in that foveola, we have a vascular free zone where the blood vessels themselves are removed from the path of light striking the photoreceptor. Not only are the blood vessels removed, but the neural elements of the retina themselves are pulled apart to the side, allowing for nearly a direct access of photons to the photoreceptors and their outer segments where the photopigments reside. So, although light can pass through these neural elements without too much of a decrement, evidently in the center of the fovea there's some additional cost that is invested to clear away those neural elements, allowing for maximal efficiency of phototransduction. Now, as this illustration should make clear, this is a very important part of the retina for vision. This is a part of a retina that can degenerate with age and with certain genetic and perhaps dietary or environmental factors that interact with our genetic constitution. And what I'm referring to is the degeneration of this part of the retina that's also called the macula. That means spot or yellow spot, macula lutea. So the macula is at the center of our retina, and the fovea is at the center of the macula. So when this degenerates, we lose the ability to see right in this central part of vision. And if I can return to that scene that I showed you earlier, this beautiful scene in Lake Louise in Alberta, Canada, if the viewer now were to have macular degeneration, they might lose their capacity to see exactly what they might be interested in detecting. If they wanted to look at our faces, it would be very frustrating because exactly their direction of gaze is what would be most affected with macular degeneration.